Hello, I'm Matthew Guy, and I will be guiding you through the most current issue of the journal Cell, as well as highlighting its effectiveness in facilitating the function of a pretty unique discourse community. So, Cell is a peer-reviewed scientific journal that publishes research papers encompassing a pretty wide spectrum of biological disciplines. Article topics include cell biology, microbiology, molecular biology, genetics, biophysics, biochemistry, immunology, medicine, and pretty much anything and everything that has to do with the cell. The goal of the journal is to provide a platform for the presentation of high-quality, cutting-edge research with the purpose of enhancing the knowledge, awareness, and congruity of this multidisciplinary discourse community. So, as indicated by the variety of article topics that are published in this journal, the discourse community that's involved in its composition include a multitude of scientific disciplines, which are all united by the common goal of understanding the most fundamental unit of life, the cell. Let's take a look at the overall structure of the journal. We can see that this particular issue has 22 articles, which have been authored by a variety of different professionals, presented in varying genres, and they have been partitioned into these different sections that we can see here. We have the leading edge select, bench to bedside, voices, previews, the review, the articles, which comprise the bulk of the journal, and then finally down at the bottom, we have the snapshot. So first we have the leading edge select, one cell in a million. The introductory article is an essay, and it's pretty accessible. There's not a lot of specialized community-specific vocabulary and terminology in here. It's pretty easy reading, and it serves as kind of like a warm-up for the denser material contained in the later sections of the journal. The author tries to grab your attention and spark your interest by highlighting a new biological visualization technique called memoir. And then down at the bottom, he presents a few hypothetical scenarios where the technique might find future application. So heading back to the journal's main page, the next section is the bench to bedside, splicing correcting therapy for spinal muscular atrophy. And this is intended to be some leisurely bedtime or bathroom reading for the more scientifically inclined. But anyways, we have a brief summary paragraph here, and if we click on download PDF, we get this nice document, which includes a cartoon illustrating the concepts of the therapy. We get some terms and nomenclature over here. We have these big bold statistics. And down at the bottom, we have a timeline of the major developments in the history of spinal muscular atrophy. Of interesting note, in 2011, ISIS was involved in phase 1 clinical trials. Pretty neat. Next we have voices, and I think this section is pretty cool. In it, we have these casual paragraphs authored by a handful of different researchers, and this section affords them an opportunity to voice their opinion and highlight some issues in developmental biology that they think are particularly interesting or important. The next section is the previews, which contain a few brief articles that summarize some ongoing cutting-edge research. We have a page or two explaining the research topic, as well as this cartoon, which aids with visualization and context. And if we actually take a closer look at the text, we can see there's a lot of unexplained lexis in here, which indicates that this was written for a specific audience, and the author assumes you have the appropriate knowledge and experience to understand it. Next is the review. Now, the review is a very in-depth and thorough article intended to provide a baseline of understanding for the community on a select topic, which in this case is RAS proteins and their regulators. This article has 3 different authors, and it draws this information from a ton of different research. As you can see all the way down at the bottom in the references section, there's a lot of citation going on here. The article also contains some cartoons and 3D models, as well as relevant charts and graphs to support and reinforce the information in the text. Okay, now we're at the articles. The article section contains original research reports and is really the meat and potatoes of the journal. Let's take a look at one in a little more depth. This article is written as a classic scientific report. It contains massive amounts of data and community-specific lexis, and it's really only accessible to members of the discourse community. There's an abundance of cartoons, images, graphs, tables, 3D models, which are all there to support the research. The scientific report is the community's primary platform for the presentation of new findings based on original research. It's typically broken down into several subsections: the summary, the introduction, the results, the discussion, where is it? And then the methods. So let's briefly talk about the purpose of each one of these sections. The summary is pretty self-explanatory. It summarizes the research. The introduction is where the authors give a little background information on their research topic and the problem they're trying to solve. The results section provides very detailed explanations of the experimental findings along with all supporting data. The discussion section is where the authors elaborate on their experimental results and have a chance to talk about the implications of the results and what it means to them and to the community. And finally, the methods section is where they describe the techniques they use in their experimentation and research. So to demonstrate the difference in accessibility between one of the opening articles and one of the research papers, I copied the text from them into Voyant Tools to get an idea of the lexis that was being used in each. So in the first article, One Cell in a Million, this is the dominant vocabulary. You can see it's nothing too crazy. The et and all here are the result of all the citation that's going on in the article. And if we take a look at the analysis from the research paper, there's way more lexis and specialized terminology in here. We've got RAS, MRAS, KRAS, NRAS, HRAS, KRAS4B, PI3K, RAF, and this represents the dominant vocabulary that's contained within the article. So that just tells you if you don't understand this terminology, you're not going to understand the article. Finally, at the bottom of the page, we have the snapshot. The snapshot is where the journal provides the community with new definitions in Lexis that have arisen due to new developments in a particular field of research. In this case, it is the emerging field of bacterial biofilm electrophysiology. Let's take a look. We have a summary paragraph here, and if we click Download PDF, we get this sideways cartoon that is putting into context some of the new concepts and terminology. And then down here at the bottom, we have a more in-depth explanation of this new emerging field. Now, the journal also does provide a mechanism of feedback and communication within the discourse community. If we click on any one of the articles, at the top we get this list of names of all the researchers involved in the article. And if we click on one of the names, we get this pop-up providing more information on the individual. We have their department, their university, their email address, as well as some other research articles that they were involved in. So overall, I think this journal provides an excellent example of how a multidisciplinary discourse community functions. And that is it for my guide to Volume 170, Issue 1 of Cell Journal. Thank you.